Welcome to the Practical Statistical Reasoning in Clinical Trials for Non-Statisticians conference call. My name is John and I'll be your operator for today's call. At this time, all participants are in listen-only mode. Later, we will conduct a question-and-answer session. Please note that this conference is being recorded. I will now turn the call over to Tracy Williams. You may begin, Tracy.
Good afternoon, CTN members and affiliates. Thank you for participating on this webinar. We have Drs. Paul Joaquin and Abigail Matthews here, and I'll give them an opportunity in a moment to give you a brief description of what they do and their affiliation with the CTN, and give an overview of the discussion and head on into it. So I turn this over to Abigail Matthews.
Hello, everyone. Again, my name is Abigail Matthews, or Abby. I am a biostatistician at the MS Corporation, and we are the Data and Statistics Center, the current Data and Statistics Center for NIDA's CTN. I'm going to be co-presenting with Paul Joaquin, who is the Senior Mathematical Statistician at NIDA's Center for the Clinical Trials Network. And what we're going to talk about in today's webinar is just kind of, first we'll go through an introduction, what do the biostatisticians do in a clinical trial? We'll go over some basic concepts of trial design. And because that's a substantive topic, we'll have a Q&A period after that. Then we'll go into the analysis plan, interim monitoring, those sorts of issues. And again, another brief question and answer period, followed by, again, a very substantive section on primary analysis. Different analytic issues to think about when you're designing your study, when you're implementing it, et cetera. And we'll briefly discuss subgroup analyses. And then finally, we'll have a lengthier question and answer period, and we hope for a large discussion amongst all the group members. I am told that if you do have a question and it is not a question and answer period, please feel free to type it in, and we have people monitoring it, and then we can discuss it when we open it up for questions. So the goal for today is really to improve the communication and the collaboration between researchers and biostatisticians. This is vital for public health and for research in general, is to be able to communicate between the actual researchers and investigators, clinicians, site staff, and the statisticians. So what we're going to also talk about is, like I said, how the biostatistician functions in the life cycle of a clinical trial, and just present some very basic statistical concepts. Again, as in our abstract, we said there will be no technical information and no formulas, and we have no Greek letters. So I hope people appreciate that we removed all our Greek letters. So I'm going to show a couple videos. This first one is hopefully what I think some people have experienced with statisticians and how very frustrating we can be. So take a look.
She got a score of 35. What does this mean?
A score of 35 and 2 points better than a score of 33 and 2 points worse than a score of 37.
What does this mean?
I am confident that a score of 37 is higher than a score of 35, 2 points higher. So, of course, that's not taking into account measurement errors.
What does a score of 35 tell me about my patient?
I have used modern psychometric methods to develop my scale. If you had used a scale that used a classical test theory approach, you would not have been using the state-of-the-art methods for measuring health outcomes.
Can you tell me what a score of 35 means in the context of my patient?
Modern psychometric methods place both people and items on the same mathematical continuum. This is better than CTN.
So this goes on with the statistician being completely unresponsive and unhelpful to the question by the clinician of, what does this number actually mean? Now we have the flip side of this communication issue, where us biostatisticians can occasionally feel that we are not being able to communicate with our
researchers. I have a study, and I need to know how many patients I need. I think I only needed 3 patients.
Okay, what kind of study is it?
I'm doing a lab study. Can I just use 3 patients?
That depends. What are you trying to show?
We always use 3 patients.
Okay, what are you trying to show?
I'm trying to show that drug A and drug B are better than only drug A.
How are you measuring if it is better?
We are looking at apoptosis. Can we just use 3 patients?
What kind of measure of apoptosis? Do you know how variable the measure is?
We always use 3 patients. That is what we have published before.
3 patients per group or in total?
Yes.
Okay, so those are examples of our lack of communication that can occur between our researchers and statisticians. And I would just like to have a social commentary that I have no idea how Paul Joaquin found these on YouTube, but apparently people care enough about this problem that they post videos on YouTube. So, let me get back to the slide. So this issue of communication, as I said, is a big one. And what we'd really like to do today is to create some common terminology and share these concepts so that we can improve the communication. Why do we have to do this? Because there are things that a biostatistician cannot do, much as we hate to admit it. We cannot propose valid research questions. We just don't know enough. We are not subject matter experts. And while we think, we have certain ideas about clinically important things and we are specialized, obviously we are not experts, which means that we cannot design a study or its statistical analysis without clinical input. And then once we have our study results, the biostatistician cannot really interpret them because we cannot place them in a clinical context. As the first video showed, we can't say what that means for a patient or a practicing physician. But at the same time, investigators can't do everything by themselves either. They cannot possibly know everything there is to know about all the statistics in the world. And while many investigators are quantitatively oriented, you really need to bring in someone who is their sole specialty. And a lot of times in clinical trials, the statistical methods needed are quite complex. And so you really do need a statistician to understand all the little nuances of different statistics and methods and the assumptions, and so you want to have one. Similarly, it's vital in terms of interpreting the results to have a statistician working with you. And so essentially what I'm trying to get at is that neither of us can do our job if we do not work together and if we do not communicate efficiently. So what exactly does a biostatistician do in the cycle of a clinical trial? The first thing before the study is we work with investigators on the trial design. We help in defining the precise primary outcome measure. Of course, we will conduct the sample size and power calculations. And generally, we will, particularly in CTN, we will write certain sections of the protocol. The one issue that is near and dear to my heart is that the statistician has to ensure that the design of the study, the way the data is collected, must be such that the research question of interest can be answered. I was an intern at the Dana-Farber Cancer Institute, very high-level research. They conducted a survey of breast cancer patients regarding tamoxifen. They brought me the data, said, this is our research question of interest, analyze the data, tell me what we got. Well, they did not create their survey such that they could answer their particular question of interest, meaning millions of dollars were wasted. So this is, again, how we can help in the trial design. Once the trial design is set, the biostatistician will aid in the development of the analysis plan. And we're going to have a whole section on the analysis plan. But briefly, you talk about the types of analyses you'll do, interim analyses. You'll specify after the trial what your primary analysis will be. And subset analyses, sensitive, all these different analyses will go into the analysis plan. Once the study gets up and running, the biostatistician will actually implement the trial monitoring and the interim analyses. So for those of you in the CTN who have seen our trial progress reports, 3 things that they focus on are recruitment. Is the study, are the sites recruiting as quickly as they need to be? Demographics, a lot of times you want to make sure your study is progressing, that you're getting enough women or minorities or any other underserved population. You also want to make sure that you're getting enough of the data you absolutely have to have, and that's your primary outcome. The other thing we do during the conduct of a trial is we prepare and present to the Data and Safety Monitoring Boards, both open and closed sessions. studies will have blinded and unblinded biostatisticians. Of course, we also, as part of these DSMB reports, will conduct the actual interim analyses. And if the study is an IND investigation that is drug, we will aid in the preparation of the annual reports. Now, in the CTN, those reports are generally done by the CCC, the Clinical Coordinating Center, but we do work with them to present the data. So now our study is over. What does the biostatistician do? Oh, I'm sorry, I'm forgetting to advance my slides here. And now it's not advanced. Okay, so now the study's over. The biostatistician, of course, this is where everyone thinks of getting the statistician involved, and that's to implement your analysis plan. So we'll create the tables and the figures and work with the clinicians to interpret the results. For the manuscript, we'll also do any additional analyses that are needed, and for the IND reports, we can help with that as well. One thing I think that's very important and is often overlooked by the researchers is that sometimes when you actually have your data at the end of a study, you realize that the existing statistical methodologies are not sufficient. And you need to engage with your statistician, and they can actually develop new approaches, new test statistics, etc., so that you can better analyze your data. And this is really vital to the process, because if you think about the type of statistics clinical trials were doing 50 years ago, it's quite different than what we do now. And without this aspect to the role of biostatisticians, the field will be completely static. So that's essentially what the biostatistician does. Right now I'm going to turn it over to Paul Joaquin, and he will discuss trial design.
Hello, I'm Paul Joaquin, and I'm going to talk about trial design. And, oops, what did I do?
To internet, yeah. And right, to go to the left, one to the left,
Sorry about that, I clicked on the wrong, oh my, I did more than I thought I did by just one click.
It's quite all right. Just give us a second. We'll get back in.
Okay.
All right.
Better put the arrow on the next.
There we go.
Okay. So what I thought we'll cover is basic designs. First, an overview of basic designs. I'm going to talk a little bit about the primary endpoint, also called primary outcome measure, and then talk a little bit about sample size and power analysis. So basic designs, and there is what's called a superiority trial, and this is what most of our trials in the CTN are. There are superiority design. And superiority design, basically the hypothesis, the clinical hypothesis, is that the experimental treatment is more effective than the control treatment. So when would we do a superiority trial? Well, for example, we want to test whether a new medication is superior to placebo. We want to test whether a new treatment is superior to treatment as usual. Or we want to test whether a medication is superior to psychotherapy. So in statistical language, we're basically testing the null hypothesis that the experimental minus the control is 0, and the alternative hypothesis that it's not 0. So this is fancy stuff to say basically that the null hypothesis that the experimental and the control are similar, are the same, and the alternative that they are different. And you may want to say, well, you may think, we're talking superiority on one hand, but here it looks, the slide looks like it's a two-sided. So what's going on? It sounds like it's contradicting each other. Well, basically, we start, we really think that the experimental treatment is going to be superior, but the test is two-sided. So technically, we're saying we really don't know which direction the superiority will go, but practically, we believe that the experimental treatment will be superior to the control. And we want to just basically start with a neutral expectation. So we expect and hope to reject the null hypothesis in favor of the alternative. And in graphical terms, this is how it looks. And I'm going to be showing you sort of confidence intervals, 95% confidence interval around the treatment effect, what we call the treatment effect, which is the difference between experimental and control. So high numbers are good. Things that when it's on the right, the experimental is better than the control. So high numbers are good outcome. And of course, we have the vertical line, which is 0. So when the confidence interval is completely to the right of 0, we have a superior experimental treatment. When it is completely to the left, we have basically an inferior. The control is better than experimental. And when the confidence interval covers 0, we basically have an inconclusive result. So these are the possible outcomes that we can get from a superiority trial. Now I'm going to talk about non-inferiority. The clinical hypothesis in a non-inferiority trial, and we do in the CTN, we do very few of those, almost none, but they are done by pharmaceutical companies and other research centers. So why would one, the clinical hypothesis is that the experimental treatment is not less effective than the control treatment. So what does this all mean, and when do we want to do a non-inferiority trial? So for example, we want to test whether a medication is not worse than an already FDA-approved medication. Or the experimental treatment has a clear advantage. Let's say the control treatment is a $100 pill that is known to be efficacious and is FDA approved, but the experimental treatment is a $10 pill that we think is no worse than the control treatment. So we just want to find out whether it is not less effective than the control treatment. In clinical language and in statistical language, we basically say the null hypothesis is that the experimental minus control, the difference between the two is less than some quantity, which is called the non-inferiority margin. So basically, in English, what we're saying is that if the difference is small, we're gonna call it. If the experimental is less effective than the control by a small amount, which is m, within that small amount, we're going to say it's non-inferior. But if the difference is bigger than that small amount, we can't say that it's non-inferior. So again, we expect and hope to reject H0 in favor of H1. And similarly, from a graphical perspective, now we have the vertical line, which is 0, and we have the margin, which is to the left of 0. And basically what we're saying is if you've got a confidence interval that's completely to the right of 0, you basically have a superior experimental treatment. But if you've done a non-inferior trial, the technical result is that it is not inferior. If the confidence interval is completely to the left, you've got an inferior experimental treatment. Now, if it is to the right of this margin, non-inferiority margin, basically we're saying that the difference is small and we can consider the experimental treatment as non-inferior. And when it covers the margin or the 0, then it's inconclusive. Now, here I put non-inferior with a question mark. It's non-inferior. Technically, it's non-inferior because the whole difference is less than the margin. But note that it is also completely to the left of 0. And if it crosses the margin, then it's inconclusive. So these are the possible results that you can get from a non-inferiority trial. And then we have the equivalence, which is the third basic design I'm going to talk about. And basically what it says is you've got two treatments that are just as effective as each, the experimental is as effective as the control. And why do we want to do such a thing? Well, perhaps one costs less than the other. Perhaps one has fewer side effects than the other. The experimental and the control have different advantages and disadvantages. Perhaps one medication is injected and lasts 30 days, and the other is a pill that should be taken every day. So you want to show how they basically have the same effectiveness. And the statistical hypothesis is, looks pretty fancy stuff, but it basically says, if you've got the treatment effect, the difference between the two is between these two margins, these two numbers, then we're going to call them as equivalent. So let me show you in the graphic. Now you have a minus and a plus m. That's the margin on both sides. And you've got the difference of 0, and you're going to look again at confidence intervals. So if the confidence interval is completely inside the two margins, then we say they're equivalent. The two treatments are equivalent. And these two are also equivalent, even though they are completely to the right and to the left of 0. Now, of course, if it is to the right of the plus n, the positive margin, it is superior. If it is to the left of the minus m, it is inferior. And if it crosses those lines, it is inconclusive. It's also inconclusive if it crosses the margins line. So these are the possible results you can get. So that's a kind of a very quick overview over the basic design. Let me talk about now the primary outcome measure, extremely important in a trial. And the very first thing I want to say is that it has to be clinically meaningful. And one test, I think a test of whether a trial has a clinically meaningful primary outcome, this is what I think of the story, is you are in a social gathering at a professional association meeting, and there is the evening cocktail social gathering, and you meet a colleague, and you say to this colleague, I'm really excited, I just completed the trial, and we got a positive result. And the colleague says, oh yeah, what did you find? And you say, well, medication X is better than placebo. And the colleague says, better in what sense? And here's the test. If you can explain in simple English what you mean by better, then you have a clinically meaningful primary outcome. So, for example, you could say among those who took the medication, fewer got the disease. Or among those who took the medication, time to death was prolonged by 2 years. Or they had a better overall quality of life. These are all good, clinically meaningful, easy to explain primary outcomes. But if you have to explain some convoluted composite of 5 or 6 criteria that defines the primary outcome, and you can't even remember all the criteria, that's going to be very difficult, and that's an indication of a poor primary outcome. And then you have simple versus composite, and simple means, like I was saying, is the number of days of use or days to relapse. A composite is when you say, oh, well, it's really a combination of days of use and number of visits attended and a score on the quality of life, and there is some mathematical formula that combines all these. This is composite. And composite, in my opinion, is a very bad idea. It's because you don't know really what you got. So Stephen Sand wrote on about 3 deadly sins in measuring clinical trial outcomes. And I just want to go over those. This is one that I haven't seen in the CTN at all, which is treating ordinal data as categorical. So you have ordinal data like which has have an order. So you have a low, medium, and high, and you treat them as categorical, meaning like gender, male, female, or white, African American, and Hispanic, without any particular order. So when you treat outcome that is ordinal and you treat it as categorical, that's a really no-no. And like I said, I've never seen that done in the CTN, so I don't consider it as a big issue. But this one I have seen, and it's creating dichotomies from continuous data, and that's what Stephen Sen calls a deadly sin, which means, and we have done that. And actually, 2 weeks ago we had a PRB meeting, and their comment was exactly that. Why are you dichotomizing a continuous variable? Why don't you just use the continuous variable and keep it continuous? So don't make it as a yes-no, keep it as a continuous measure, because a yes-no automatically implies that you're going to have to decide to determine a threshold. And any observation just to the right, to the left of that threshold would be a yes, and to the left would be a no, and that's kind of very artificial. And another no-no is using change from baseline. There's no need to use as the predictor variable, the y variable, the variable on the left of the model, as a change from baseline. Use the value itself, and if you want to control for baseline, just put it as a covariate in the model. So now I want to go over sample size and power analysis. It's a lot of stuff to cover, so I'm going to kind of go a little bit quickly on that. So, and I'm going to talk about what the biostatistician needs and why. So the clear things is the number of treatment groups, superiority or non-inferiority or equivalence, what kind of trial do we have, and whether it is one-sided or two-sided. And like I said, we usually typically use two-sided. So these are, the first three bullets are kind of pretty straightforward. But then there's the expected dropout rate. And, well, if we expect a high dropout rate, we need to increase the sample size. And if we expect a low dropout rate, we don't need as high a sample size. And basically, we increase the sample size to account for the expected amount of missing data in the primary analysis. So that's a very, very quick explanation, even though there's a lot more to it. Another thing that the biostatistician needs is the smallest meaningful clinical difference to detect. What is it that, the difference that really makes clinical sense, that really makes a difference? And what is the smallest such difference we want to detect? And if the difference we want to detect is big, well, then the sample size can be small. But if we really want our trial to find a very small treatment effect, then we have to increase the sample size. Then there is it depends on the alpha, or the chance of type 1 error. Typically, we use the 5%. This is kind of a general agreed on value. Nothing magic about it, but just a reminder of what the alpha is. The non-technical definition is that it's the chance of concluding that the experimental treatment is more effective when in fact it is not. So it's an error. We make an error. We say it is more effective when in fact it is not. In technical definition is the probability of rejecting H0 when H0 is true. But here's what's interesting is, talking about the different perspectives, the FDA is particularly concerned about this because it doesn't want to approve a medication that's not effective. And that's making the type 1 error. So FDA wants to control for this, and basically, I think dictates that, pharmaceutical company uses a 5% alpha. But the pharmaceutical company is not that concerned about that. I mean, the conclusion is wrong and it says the medication is effective, I don't think the pharmaceutical company is really going to complain about that. So very different perspective on the same concept. So the bottom line is that the most commonly used value of alpha is 0.05 for a two-sided. And if the alpha goes up, sample size goes down, which means if you allow yourself to make an error, then you don't have to have a big sample size. But if you really want to control and have a very small chance of making this kind of error, you have to increase the sample size. And then there's power. The power to detect an effect, which is, again, usually 80% or 90%. The non-technical definition of power is the chance of concluding that the experimental treatment is more effective when, in fact, it is. So that's why we say the power, the probability to detect an actually effective treatment. The technical definition is that the probability of rejecting H0 when H0 is false. And again, I want to talk about those two perspectives. The FDA is not particularly concerned about power because it knows that the pharmaceutical company is going to be concerned about it. And because the pharmaceutical company wants to make sure that if it has in its hands an effective treatment, it wants to make sure that its trial is going to detect that. It doesn't want to miss something that was actually effective. So the bottom line is that the most commonly used value for power, at least in the CTN, has been somewhere between 0.8 and 0.9. It doesn't have to be, but that's typically what we use. And if the power goes up, the sample size goes up. If we want to be able to detect a difference, we have to increase the sample size. And if we don't particularly are concerned about it, then we can keep a small sample size. The variability of the primary outcome is also something we need about for power calculation and sample size calculation. If the primary outcome varies a lot, we need a high sample size, and if it varies a little bit, we can do with a small sample size. And I'm going to leave it at that. If there are questions, I'll be happy to answer. And finally, the last thing is the correlation between measurements within the same cluster. It's also called the intra-class correlation, or ICC. So let me give you an illustration of this. This is from Wikipedia, and it's basically what you see is the data value is on the vertical axis, and the group number or cluster number is on the horizontal. So each line is a cluster. Now, what's a cluster? A cluster can be repeated measure on a participant. A cluster can be several participants in a group therapy under the same counselor. A cluster can be a class in a school. A cluster can be a hospital. So it's a unit where the observations from that unit may be correlated. So in this particular graph, you see the different values for within each cluster. So for number 1, the values, you can see that they are around, what is it, minus 0.2, something like that, and et cetera for each cluster. And you can see in this graph that they tend to be together. That's an indication of a high interclass correlation. And this is an illustration of a low interclass correlation, where you can see within a cluster, the data values are all over the place, within a cluster. So if the correlation is high, you need a high sample size. If the correlation goes down, the sample size goes down. And this may be a little bit puzzling because you say, well, I thought correlation is a good thing. A high correlation is a good thing. And here it seems like I'm being penalized for a high correlation. When the correlation is high, the sample size is high. Well, here the interpretation is that when the correlation is high, each observation brings little information because they're correlated. If the correlation is low, it means that each observation brings a very different piece of information. And it basically takes, it's almost like increasing the sample size. So a low correlation is actually better for the sample size. And basically I want to show this slide, which here what I did is I put all what this biostatistician needs, and I kind of color-coded it. The white is kind of pretty easy to determine. The yellow is a little bit harder to determine, and the red is really hard to determine by the clinician. And but you know, we do in the CTN do for several values and see the power for different combination of values. And the sort of one final note about sample size is that cost, which has nothing to do with biostat, is most often a key factor in the final decision on sample size. And I think this is the time where we're going to pause for questions.
John?
Thank you. If you do have a question, press star then 1 on your touch-tone phone. Once again, if there are any questions, press star then 1 on your touch-tone phone. And at this time, I have no audio questions.
And we'd like to also remind participants that feel free, if you're a little shy, to chime in, that you can use the Q&A feature in the dashboard. We can continue.
Okay, I think we'll get back to the presentations then. So this section, we're going to talk about the analysis plan, what goes into it, and what the issues are that you have to think about. So the purpose of the analysis plan is to specify your analytic methods a priori. I'll probably be saying this many more times in the rest of the webinar, but it's incredibly important to do all of these things ahead of time. And that really is one of the sole functions of the analysis plan, because your analyses cannot be data-driven. You can't say, well, now that I have my data, I look in these 10 test statistics, all of them are negative, but this one is highly significant. I'm going to report that. We cannot do that. And so by having our analysis plan set before the study is completed, we kind of remove the criticism that these are data-driven analyses. So for the CTN, how we do things there is that the analysis plan must be finalized before data lock. So if some of you are on a lead team and you're dying for your data, but you haven't seen your analysis plan, you got to get on your statistician to send it to you so everyone can sign off. And that's, you know, the other thing about the analysis plan in the CTN is that while it is developed by the statisticians at the Data and Statistics Center, the lead team has input, and they must also sign off on it. So this is a group effort. So there's kind of quite a few components to an analysis plan. You have to specify the population that you're going to look at. You also want to specify for your primary outcome measure the statistical test you'll be using or a model, whatever you decide. There's some issues you have to think about beforehand, and that's, do you have multiple comparisons? If you do, how are you going to handle them? How are you going to deal with missing data, as well as outliers? And there's additional analyses that must be specified in the analysis plan, and those are interim analyses, such as sample size re-estimation or interim efficacy analyses. You also want to talk about different sensitivity analyses you'll be doing. I'll get to examples of some of those. And any secondary outcomes or secondary analyses of your primary outcome that you'll be doing should be specified beforehand in your analysis plan. And then also subgroup analyses should be determined before the study is completed. So in terms of the population to be analyzed, there are really kind of two different cohorts people can look at. One is called the intent to treat, or ITT, and this is analyzing every single participant that was randomized. Some of you may have heard of the phrase once randomized, analyze, or once randomized, always analyze. There's something that is not completely intuitive about this population, and that is that even if a participant has no data after randomization, they are still considered part. They can still be considered part of this ITT population. So that's something to think about. In the CTN, we generally do ITT analyses. But there's also per protocol analyses, or PT. This is where you, again, a priori, in your analysis plan, you specify a particular subset of randomized participants that you will be analyzing. So in a medication study, you may elect to only analyze participants who had a certain level of exposure to your medication. Say they took at least 80% of their pills. In the CTN, it's possible you may want to restrict your analyses to only participants who attended at least 50% of their treatment as usual group sessions. So those are the two kind of general populations that you can think about. Then you also have to specify in your analysis plan the statistical tests or models that you'll be using. And for testing, you want to think about which particular test statistic you're going to use. And this, again, involves collaboration between the statistician and the investigator, because there are certain assumptions that must be thought about, involved with certain statistics that may not be clinically relevant or appropriate. Again, another one that has to involve the investigator is what time points will be looked at, and also what exactly you want to use to measure your treatment effect. You have to think about this. You don't want to choose, well, I want to know what it takes to get their rate of abstinence from 46% to 47%. You know, a patient probably doesn't care if they're going from 46 to 47%. You want to use a measure of treatment effect that is relevant. If you decide to go into the modeling situation, you can have these incredibly complicated models, but you absolutely must have a parameter or parameters that can be used to test your primary outcome and the corresponding hypothesis. If you're throwing in 10 parameters to capture treatment effect, you have to specify, again, a priori, what the null hypothesis is in relation to those parameters in the model. So some models that people use are longitudinal or repeated measures, for example, in the CTN, where you're, say, monitoring abstinence over 10 weeks of treatment. You can also look only at a single time point. One thing, the team task force recommended was looking at abstinence in the last 4 weeks of treatment. That's another modeling thing. You also have to think about what factors are going to be included in your model. And these, again, you're not saying how they will be entered into your model necessarily in the analysis plan, but what they will be. Are you going to consider a time by treatment interaction? Are you going to look at covariates such as age, race, socioeconomic status? Are you going to include the stratification factors for randomization? One thing that really could actually be an entirely separate webinar is side effects. And this is very important, particularly in the CTN, because of the treatment as usual component. We have the potential for substantial side effects. So all of these things must be thought about beforehand and specified in the analysis plan. Now, the other issue I mentioned was you have to identify if you have multiple comparisons, and if you do, then what are you going to do about it? Now, the reason this is important is because, as Paul was referring earlier, we have this false positive rate or type 1 error rate, and usually we set it at 5%. So that means if we do 100 statistical tests, 5% of them, so 5 out of those 100 will be statistically significant purely by chance. So just because, if we're doing 1 million subgroup analyses and one of them is significant, that does not mean that there truly is an effect. So you have to take this into consideration when interpreting your results. So when we do have this, I can think of some examples. If you have co-primary outcomes, I know in CTN 44 we have that, or if you have multiple treatment comparisons, for example, having 2 different doses of a drug versus placebo, then you again have a multiple comparisons issue. Or if you're not interested in a longitudinal model, but you still want to look at treatment effect at 2 different time points. So once you know that you have it, how do you handle it? The most common way is the Bonferroni correction, and it's incredibly appealing because it is very simple, but it is very important to remember that it is also extremely conservative. Essentially what the approach does is it splits that overall type 1 error rate, that 0.05, between each of your test statistics. So if you have 2 tests that you're performing, you split it the 0.05 in 2, and you compare each of your p-values to 0.025. Now, there are many other procedures for adjusting for multiple comparisons. This is just one of them. One of the major issues that we statisticians have to think about, particularly in addiction research, is missing data. Now, Paul Joaquin did have a publication last year where he looked at the first 24 CTN trials from 2001 to 2010 and saw that the rate of missingness for the primary outcome measure varied in those studies from 2%, which is phenomenally low, to 60%, which is incredibly high. So if you had, let's say, 200 participants and you had 60% missing, you can think about that you only had 120 participants. I'm sorry, I can't even do that math in my head. 80 participants with data that you could analyze. So because this is such an issue in the addiction research, again, this could be an entirely separate webinar, is how you handle this. But there are various methods, some are very simple, and we've seen this done in the CTN, which is when you're looking at abstinence, you impute missing abstinence as, say, youth. But then there's also very complex methods, like pattern mixture models, which I'm not even incredibly familiar with. It's so complex. But I think one thing I would like everyone to take home today is the 3 types of missing data, and this is what we have to think about in developing our analysis plan. The first, the simplest, which is called missing completely at random, or MCAR, or MCAR. And this situation is where the fact that the observation is missing is completely random. So essentially, the participant did not show up to their study visit because their car broke down, and that's the only reason. Otherwise, they would have been there. Slightly more complex is what we call missing at random, or MAR. And here, the unobserved data, the data that's missing, can actually be explained by data that you have already observed. So whether that's covariates or in a longitudinal setting, if you already know that the person's been abstinent for the first 10 weeks, you absolutely know they're going to be abstinent that last week. The nice thing about the MAR setting, when we have this, is that a lot of the commonly used statistical methods are valid when the data looks it is missing at random. However, in addiction research in particular, we generally have the setting of missing not at random, or MNAR. And here, the observed data cannot be explained by data that we have actually collected. So, in our CTN studies, it's quite common. You think we can safely assume that if a participant does not attend a study visit, it's because they were out on the street and using. However, in my studies, I've also had participants say, well, it's not fair, I can't come in to my study visit because I'm doing so well, I'm sober, I have a good job. So in this case, you can't assume that it's youth or non-youth. You just don't know. The drawback to this, and this is, again, why you have to get your statisticians thinking way from the start, is that standard statistical methods cannot be used in this case, and you need something more sophisticated. And all these things have to be specified in your analysis plan. Outliers, just a nice definition from Mosulsky, says that an outlier is a value that is so far from the others that it appears to have come from a different population. And what he mentions, and this is important to know, is that the presence of outliers in your data set can actually invalidate your statistical analyses. One thing that people do is they will run their analyses with the outliers and then separately without and see if the results change. There are also statistical methods out there to identify what is an outlier, but we won't go through that today. For your interim analyses that have to go into your analysis plan, you want to make sure that you specify the type of interim analysis. that you're going to be performing, whether it's sample size re-estimation, futility, or efficacy. You don't really want to do all of them. You also want to specify when these will occur. It cannot be willy-nilly. You cannot say that we will do sample size re-estimation in the middle of the study. So, for example, I think in most of our studies in the CTN, what we've done is we will do the sample size re-estimation when approximately half, we can't always guarantee that it's exactly half, of the participants have completed the active treatment phase, so they have their primary outcome data collected. You also have to specify the frequency, how often you will be doing these analyses. Generally, sample size estimation, you don't do them repeatedly, but sometimes efficacy people will look quite often. For example, if you know your DSMB is going to be meeting every 6 months, you may decide to perform an efficacy analysis for each of those meetings. The other sets of analyses that must go into your plan are sensitivity analyses. And essentially these are, all they really are is things you do to assess how sensitive your study results are to various assumptions and methods of analysis. So one thing that we generally will look at is you want to compare different statistical methods. So you may use one model, get an answer, use a different statistical model, get another answer, and see how much agreement there is. You also want to look at the different methods of handling missing data. And this is very important, again, in addiction research. So there will always be a substantial number of sensitivity analysis for the CTN. Secondary and subgroup analyses must also go into the analysis plan. You want to, again, just make sure that you specify these things a priori. You don't have to be incredibly specific. You don't have to say, I'm going to use a t-test and blah, blah, blah. But you have to say that you're going to look at, for example, the longest stretch of abstinence for a participant. The subgroup analyses that are required of us are gender, race, and ethnicity, so these should go into your analysis plan, as well as any others that you anticipate may be informative. So that's essentially what goes into your analysis plan. The next thing that is important to discuss today is the trial monitoring and interim analyses. So for trial monitoring, I think some people have actually seen the CTN trial progress report, and they contain everything on this list except the first one. So we'll start with number 2. We do monitor. You always want to monitor regulatory compliance. Is your IRB up to date, your federal-wide assurance, all that stuff. You want to make sure the study is recruiting properly, not just overall, but within site as well, and within certain demographics. You want to make sure that you are collecting that primary outcome data. You don't want to have a substantial amount of missingness. You also obviously want to ensure that your participants are getting enough treatment. Tentative follow-up visits, while this is not primary outcome usually, this is very important. And of course you want to look at data quality. So whether that be your missing forms or missing values or data anomalies. The first one on this list is actually, in terms of the CTN, it's really monitored by the CCC, the Clinical Coordinating Center. And it is not included in our KPIs because this safety information on adverse events and serious adverse events is blinded information, so we don't present that to the group at large. However, it is monitored on a daily basis. So those are the essentials of trial monitoring. In terms of interim analyses, what I'm referring to is essentially when we look at the data during the conduct of the trial, before data lock. And your objectives are essentially to modify the study if you must, such as with a sample size re-estimation, or determine whether the study needs to be concluded prematurely. Why would you want to do that? Well, it's possible that you can demonstrate one treatment arm is substantially less safe or unsafe, and you do not want to expose participants to that treatment. Similarly, you do not want to assign participants to a treatment that is clearly ineffective or substantially less effective or efficacious. The last reason that I I think of, and I hope none of us have had to deal with this or that we ever have to, is if there are insurmountable logistical issues. Sometimes until you actually sit down and analyze your data, you don't realize the data quality. For example, you can look at everything looks good, all the values are answered, you have no missing data, but when you look at it and you analyze it, things just don't make sense. And the question is, can that be remedied, or does the study literally have to stop? So in general today, I'm just going to talk about 4 types of interim analysis. So there's the sample size re-estimation, efficacy, futility, and harm. Now, the sample size re-estimation, why would we want to do this? When we're designing a study, generally there is little information known a priori. And if you recall Paul's slide where he had the things the biostatistician needs, there were several things in red, and those are the ones that are very hard to determine. And so if you have very little certainty in your initial power calculations and the assumptions used and the parameter estimates used, you're probably going to want to do a sample size re-estimation. So there's different ways to do this. I'll just give one example. Say we have a 2-arm study. One is placebo, one is active treatment. What you could do is simply analyze the placebo arm. So you're not actually looking at your active treatment arm. You estimate all your parameter estimates that you had used in your power calculation, and then determine the sample size needed to detect that clinically meaningful effect. And that clinically meaningful effect is the same as what it was in the original calculation. And you may have to up your sample size, or if you see that it's substantially lower than your target sample size, you may decide to shut down the study. The nice thing about such an approach is that you're not estimating the treatment effect, so there is no impact on your overall type 1 error rate. This is not the case with efficacy and interim analyses. And with these types of analyses, what you're doing is you want to identify, is one treatment arm clearly inferior to the other, or clearly superior? So how you would do this in general is that you take your final data analysis that you would be doing on your primary outcome, and you implement it, obviously, during the conduct of the study. And then there's these certain what we call stopping rules, and it's based on the results you see from that final analysis approach, do you stop or not stop? And there's different stopping rules. The advantages of an efficacy analysis is that you can even not necessarily terminate a study, but you can drop a treatment arm. For example, if you have 3 and one dose is clearly not working, you can simply drop that arm and then only have 2. And obviously, doing this, it prevents exposure of participants to a clearly ineffective treatment or substantially less effective. Now, the disadvantage is, one, it requires unblinding. So you're having your statistician actually look at individual treatment assignments. The other is that because you are looking at your primary outcome in the middle of the study, we call that kind of a look. And so you're going to have multiple looks, meaning multiple comparisons, meaning you have to adjust your overall type 1 error rate. Now, futility, the question is a little bit different. It's not is one treatment arm significantly better than the other. The question when you do a futility analysis is, given the data that we have collected thus far, is there clear evidence that there is no treatment difference between the 2 conditions? So, again, it's a little bit different. And here, essentially how you do it is you compute what's called the conditional power. And the conditional power is the probability of detecting a true treatment effect. So there really is one, the probability of detecting it given the data that you've collected thus far. Now, a priori, so again, in your analysis plan, you must specify what is an unacceptable conditional power. So I think in some CTN studies we've used 20%. That's the value we used. Now, for harm, there's different ways you can do an interim analysis of harm, but essentially you're just asking, is one treatment arm unsafe or significantly less safe than the other arm, such that you do not want to expose patients to that? So one thing you can do is determine acceptable limits for rates of adverse events or serious adverse events and see similarly, you can test whether the frequency and or type or severity of these adverse events differs across two treatment arms. Now, because you're not actually looking at your primary outcome, there's no impact on doing these kind of tests of safety information, so that's nice. However, it does require unblinding. You are going to look at treatment arms if you're testing that the two treatment, that the AEs and SAEs differ between the two treatment arms. So I'm going to open it up for questions right now. If there's any questions on the analysis plans or trial monitoring or types of interim analyses, we have a few minutes for those questions.
And once again, if you do have a question, press star then 1 on your touch-tone phone. I did have a question from Therese Killeen. Please go ahead.
Yes, I wanted to ask about when the investigators for a CTN trial have a publication plan and full of secondary analyses. So you're saying that should be established prior to a priori?
Yes, as much as possible, all of your secondary and subgroup analyses should be specified a priori. However, it is possible that you think of things during the study, but I think Paul has something he'd like to say.
I mean, it's not a black and white thing. I mean, basically, I think, to be honest, you have to put in the manuscript whether this was determined a priori or not.
Oh, okay, yeah, that makes sense.
Once you do that, I mean, the reader, if I'm a reader and I'm reading a manuscript and it says this secondary analysis was predetermined up front, I would trust it a little bit more than if the manuscript says, oh, we're basically exploring and we found this result. So it's really it's a combination of honesty on the part of the writers, the authors, and it's a question of confidence in the result on the part of the reader. The more it is predetermined, the more it is believable. Not that the result is wrong, but that the result is not just by chance you found something significant.
I see. So kind of putting it in a limitation.
Yeah.
If that's the case.
Yeah, and if you're honest, I mean, if you say, after we did these analyses, it wasn't part of our plan, but we decided, to do this analysis because we looked and it looks like we see something, and we did the modeling and we did the analysis, and we did find a significant result. Now, if a good author would also say, but keep in mind, you reader, that this was done, after the fact, and, true, it is significant, but it shouldn't be considered as the definitive result. And really, something more has to be done. More research is necessary, I guess.
Okay, thank you.
At least we know that somebody's alive out there.
We have some questions in the Q&A.
Oh, we click on the Q&A? Okay.
Okay, so we have a question from Shari Ginsburg-Labor. I'm being called in different directions. Oh my goodness.
Oh, okay. They're leaving the...
Yes.
Okay.
So
no questions.
No actual questions. But, Harry, we will get to your question later.
Once again, if you do have a question, press star then 1 on your touch-tone phone. Well, we do have a question from Nahami Ahmed. Please go ahead. Go ahead with your question. Your line is now open.
Can you hear me?
Now we can, yes.
So I just kind of had a follow-up question to that. You know, when you're doing research, sometimes things come up that you hadn't thought about necessarily before. I guess what are your kind of suggestions for at what point is this kind of data dredging?
You know, it's very tricky. I mean, you obviously don't want to ignore something that is important to look at. You don't want to neglect. It's just a very tricky balancing act. And I think what Paul's comment was is very important, is that you have to be forthright, and you have to say, you don't, in your manuscript, you don't have to say, well, we looked at 57 different analyses, and we're reporting 7 of them. I mean, I don't think you have to be that blatant. But you absolutely must acknowledge that this was not planned, and, you know, yes, data dredging, but, again, you really don't want to miss an opportunity to look at things just because they were not specified. Really, the most important thing is to specify your primary outcome beforehand and report that. I think that is absolutely vital. And, of course, you know, as you said, there are always interesting data points that you couldn't even possibly have thought of previously, and yet you're seeing them in your data. And you should be able to explore that. You just have to acknowledge it publicly, that this is what's going on.
Yeah, and think about the next person who's reading the manuscript. I mean, if let's say it wasn't pre-planned, you do the analysis, something comes up and you think of something, you do the analysis, it is significant, you publish it, and you acknowledge that, you know, this was done, you know, after the fact, but this is what you found. And it should be taken with not as a definitive answer. Now, think about the reader. Now, there is a reader out there who reads this and say, you know what, I mean, this is a hint, this is a clue. I think it's important to me. Now, this is another person. I think it's important to me, and I'm going to do, based on this possibility of a significant result, I would like to do a trial, or I would like to explore it more. And think about this next person finding a significant result. So that's going to confirm. Now we have 2 results that were significant. So I guess this is a long way of saying is don't ignore it, put it publish it, acknowledge its weaknesses, and let science, you know, as long as you're honest, then the reader knows where it comes from and how it was obtained.
Okay, thank you.
No further questions at this time.
Okay, well, now we're going to hand over to Paul, and he's going to talk about primary analyses.
Okay, so in terms of primary analysis, I mean, and it's connected to what Abby was talking about, the analysis plan. Of course, you know, the primary analysis is going to have to be consistent with the analysis plan. So there is a lot of overlap there, and there is a lot to talk about under the primary analysis. But because of time, I thought I'll cover 2 things. One is I'll make a few general key points. And then, you know, I'll focus on the interaction. What does interaction, the word interaction, mean? So let me start with some general key points. And I'm going to start with an example. This is called Anscombe's quartet. And you'll understand in a minute why it's called a quartet. But Anscombe is the name of a statistician who came up with this example. This is made-up numbers. It's not real. And he came up with this example, and you can find it on Wikipedia. It's amazing how many things statisticians find on Wikipedia. Very useful stuff. But anyways, the example is the following. We have 11 pairs of measures. So you have an X and a Y for, let's say, 11 participants. Okay? And they produce the following statistical results. So you've got the average of X, you've got the variance of X, average of Y, variance of Y, correlation between X and Y, 0.816. That's a very high correlation, particularly in the psychology field. That's very, very high. And then you have the regression line. So you do maybe we can overall mute. Okay, so then you have a regression line, and it gives you the regression line. So you say, okay, so I've got my data. I've got all the information I need, the average, the variances, the correlation, and the regression line. So I'm basically done. And I would say, wait a minute, why don't we graph this data? Okay, so we graph the data. And here's the regression line. You can see it, the white line, and you've got your 11 data points. And they basically look like what you would expect. A high correlation, a nice regression line in the middle of the points, and everything looks fine, right? Well, let's look at another possibility. So these points, the 11 points that you see now, produce the same results that you saw 2 slides ago. In other words, these points would give the exact same average of X, average of Y, correlation between X and Y, the variances are the same, the regression line is the same. And yet, when you graph the data, this is how the data points look like. Oh, okay. So here's another example. Again, these points that you see now produce the exact same table that we saw a few slides ago. So that's a very different picture. And this picture also, these 11 data points also produce the same averages, variances, correlation, and regression line. So this is where the quartet comes in. So you've got the X1, Y1, which is what you would imagine in your head when you see the table. At least that's what I would think of. But then the X2, Y2 says basically the data points are not linear. And we basically force the correlation, as you all know, is a measure of linear relationship. So by just looking at the correlation and the equation of the regression line, we missed what's really going on, which is a nonlinear relationship, which is shown in the X2, Y2 graph. In the X3, Y3 graph, we see that basically it would have been a perfect relationship through all the points except for that odd one. And that odd one is pulling the regression line towards itself. And then the fourth one, you see that there is really no relationship at all. And the only reason we got such a high correlation is because of that really odd point out there on the right, which is really an outlier, which is what Abby was talking about, an outlier, which is really completely different from the group of other points and has a big influence on the analysis. So what's the point of all this? The point is to graph, always start with a simple graph of your primary outcome. If you have a longitudinal trial, do it over time and do it by treatment group. So look at the graph of the data, because the p-values and the regression line and the regression coefficients and the averages, all these descriptive statistics are not going to give you the full picture. And a graph would really reveal things that you may not see with the descriptive statistics. The general key point number 2, which is a big deal, and it's hard for an investigator to basically be convinced of that. But the point is, if the primary research question is important, the answer, the result is important. And it's regardless of whether it is positive, negative, or not, as long as the trial was a well-conducted, well-designed clinical trial. So if it produces a null result, it's not a failed study. I've seen that it's called a failed study. It's not failed at all. It answered the question. The answer just happens to be null, that there is no difference between the two treatment arms. I mean, yes, we would love to find the cure for cancer. If we had a trial and we find the cure for cancer, this is fantastic. But the point is, even if a trial doesn't produce a significant result, a null result still advances scientific knowledge. And the way I see it is, imagine you have a list of possible treatments that you think will treat cocaine addiction. So you start with what you think is the most promising, and you get a null result. Okay, well, if it is well conducted and well designed trial, you can take this potential treatment off the list, and now you have narrowed down your list. So you are advancing scientific knowledge. Abby talked about sensitivity analysis. I think it's extremely important to do sensitivity analysis as part of the analysis and to put that in the primary manuscript. So to present the results with at least one variation of the primary analysis. So it could be, for example, a slightly modified outcome. It could be your assumptions about missing. How are you going to handle missing? And I'm going to give you an example about that. So a slight variation, a natural. If this is really the way I would do it, what would be the next way I would do it? Kind of thing. Here's an example. This is a publication from George Woody, a JAMA publication. This is on CTN 10, Bupe for Adolescents. And this is straight from the journal article. And the authors showed two graphs. One, it's the main results of the trial. But they did a sensitivity analysis, and they did the analysis two ways. They analyzed only the observed data. This is the graph on the left, and the graph on the right. The missing data was imputed as drug use. So they did it both ways, and they showed it the result. And of course, the results are not going to be identical. That's a mathematical impossibility. But what's important is, are the conclusions consistent? And if the general conclusions are consistent, then you really have strengthened the evidence of the conclusions. The general key point number 4 is, and this I've seen so many times, is to convert the statistical results to the original scale with point estimates and corresponding confidence intervals for the primary outcome and for the treatment effect, the difference between the two treatment effects, the two treatments. So sometimes the primary result is presented in terms of odds ratios, or even worse, log odds ratios. Sometimes they're presented as slope, or the intercept, or the area under the curve, or some parameter of some statistical model. But what does it mean clinically? And that's really the point I'm trying to make, is and I've seen papers, they say, oh yeah, the main result is that it's significant. Now, you don't know what it is, but it is significant. And this is the p-value. It's 0.001. And I've got a reference for this. I'm not making it up. And you have no idea, the reader has no idea what we're talking about clinically. What does it mean? So it is important to translate the statistical jargon into something meaningful. And that's really related to the point number 5, which is really to understand in simple English, not in statistical jargon, what the trial, what the results is, what the primary results mean. Now, this is a list of possible things I would go over in much more detail in terms of rejecting H0 versus not rejecting H0. P-value, we can cover, just the P-value. I've got about 15 slides just on P-value, but we don't have time, and I thought, we'll focus on interaction, and that's what I'm going to do next. So I'm going to basically, talk about interaction. So this is this is a statistical term, and it comes from a model, a statistical model, and I'm going to a little bit explain what that means. But to explain interaction, we really have to talk about those four things: the treatment effect, site effect that Abby mentioned, and then start to talk about treatment by site interaction as an example. And then I'm going to talk about quantitative and qualitative interactions. So a treatment effect is pretty straightforward. We know what it is, and I'm going to show you this graph, and we're going to show this graph repeatedly, this kind of graph. Basically, what you have on the vertical axis is abstinence. So a high number is a good number. And the gold bar is experimental treatment, and the green bar is the control treatment. And we all know what a treatment effect is. It's the difference between the experimental outcome, the outcome from the experimental group minus the outcome from the control group. That's called the treatment effect. And we do have one Greek letter, delta, in English. So this is a treatment effect. Okay, fine. Now, what is a site effect? And Abby mentioned site effect. So now we have a multi-site trial. We have the overall result on the left. We have a significant overall treatment effect. Same thing, abstinence is the scale. High numbers are good. But here, since we have a multi-site trial, we have 3 sites, A, B, and C, and these are the results from each individual site. So what is site effect? Site effect is, think about taking the average of the yellow and green bars, take the average of those two, and see if they're very different from site to site, and if they are different from the overall. If the answer is that they're not that different from each other, and they're not that different from the overall, there is no site effect. Okay? So here in this particular example, you can see that overall, the levels of abstinence in each site, sure, there is a difference between control and experimental, but overall, the average of experimental and control across the sites are basically the same. So that's no site effect. So what does the significant site effect look like? This is how it looks like. So you can see here, we do have a significant site effect. And you can see here that site B overall has much lower levels than site A. Now, why is that? And that shouldn't be any surprise, because it can come from all kinds of results. It can be that they're serving different. The sites are serving different population. It could be that the geographical location. There is a prevalence of one drug of abuse rather than the other. It could be that the counselors have a different philosophy. It could be all management of the site, budget. It could be all kinds of different reasons. But that's not really, first, it's not an odd result. And second, it's kind of a site thing in terms of what we're trying to find. A significant side effect really is not relevant, I guess, relevant to the primary conclusion. It is important to take that into account in the model, but it doesn't mess you up, in other words. So that's a side effect. And now we go into treatment by site interaction, and that's a little bit more difficult to understand, but I'm trying to put it in a picture. So here's an example where, again, same story, 3 sites overall, abstinence. But we see that there is no site effect. I mean, the general levels are the same. If you take the average of the green bar and the gold bar at each site, you pretty much get the same average. So there is no site effect. But look at the difference, the treatment effect, which is, remember, it's the white arrows, the treatment effect. The overall is significant, but if you look at the white arrows at each site, you can see that site C, the treatment effect is much larger than in site B. Now here, we're talking definitely relevance to what we're trying to analyze and what we're trying to understand. Because here now we're talking about the treatment effect, the difference being different across the sites. So still on the treatment by site interaction, there is what's called a quantitative and a qualitative interactions. And I'm going to give you an example of each. Here we have a significant overall treatment effect. That's the white arrow on the left, above overall. And we have a significant treatment by site interaction, because we can see that for site A, the treatment effect is a lot smaller than for site B and C. So we do have a treatment by site interaction. And furthermore, we say this is a quantitative treatment by site interaction. Quantitative means that it varies by quantity. So the quantity is different. The treatment effect only in quantity is different from site to site. And it's not qualitative. And then I'm going to show you an example about a qualitative treatment by site interaction. But again, it goes in the same direction. In other words, for each one of those three sites, the experimental is better than the control. By how much is the part that varies from site to site. Now we go to the qualitative treatment by site interaction. So you can see that the overall there is a significant treatment effect. But if you look site by site, you can see that for site C, actually it's the experimental that's better than it's the control that's better than the experimental. And for the other two sites, it's the other way around. So we say it's a qualitative treatment by site interaction. Now this is very important to know about. Can we say that the overall treatment effect is significant? I mean, if you look at under overall, the two bars on the left, you say, yeah, it is significant. But now that we know that really the treatment effect is not going in the same direction across the three sites, the answer is one really needs to examine each of the participating sites and try to determine what are the differences between these sites that could explain the differences in their response to treatment. And this was found in one of the CTN trials. And they found that there was a likely explanation for the different results, and that the one side that went in the opposite direction, the participants were mostly court-ordered. So what's the bottom line? The first point is that there is no major downside to including a site effect in the primary analysis. In fact, it may increase power because the site effect in your model may explain part of the variability, the variation. And that's a good thing. That increases your power. Testing for a treatment by site interaction is important. You do want to know if the treatment effect is consistently the same in the same direction across the site. And on the first point, I want to emphasize that a significant site effect, that's the first bullet point, a significant site effect is not a problem. It's not something to worry about. But a significant treatment by site interaction does affect the interpretation of the overall treatment effect and the generalizability of the conclusions. So it's very important to test for the treatment by site interaction. And now the general thinking is that, oh my God, we found a significant treatment by site interaction, we're doomed, the whole trial is a failure. Actually, no, I don't think so. Because if you have a treatment by site interaction, again, remember, a treatment by site interaction, especially a qualitative one, where one site is very different, the treatment effect in one site is very different from the other site, this could be really a gold mine. Because if you can explain why this site was different from the others, it really may shed light on some very important factors that modify treatment response. And if you explore that in more detail, you may find something really significant in terms of treatment. So yes, the first reaction is to kind of say, we've got a problem because we have an interaction, a significant interaction term, but in reality, it may be something very useful. And I'm going to stop here and turn it to Abby.
Okay, very briefly, I'm going to talk about subgroup analyses. So what exactly am I referring to when I say subgroup analyses? They're actually a particular type of secondary analysis, and they look at differences in the treatment effect among different subgroups of the randomized participants. So normally you would like to specify as much as possible of your subgroup analyses in the protocol or your analysis plan. But as we discussed previously, sometimes these subgroup analyses really only become relevant during the conduct of the study or as you're looking at the data. So while this is not preferable, it does happen. Examples that we do for subgroup analyses required by NIH are by gender, race, and ethnicity. Other common subgroup analyses focus on age group or socioeconomic status. One that I think has been done in the CTN, I'm not positive, is looking at the severity of the disease or disorder at baseline. And that's an important point to make for subgroup analyses, is that you really need to identify the subgroups based on pre-randomization information. So again, if you were doing severity of disease, it's the severity of disease at baseline. So in the CTN, you could do a subgroup analysis for participants who had a positive UDS at baseline and separately in participants who were negative. Again, because you are performing all of these analyses and these are exploratory, they are not going to be adjusted for in your multiple comparison. So you have to interpret them with a grain of salt. And that means that ideally you would keep the number of these subgroup analyses to a minimum. Generally, there's kind of two approaches to take with these subgroup analyses. One is you can perform your analysis separately in each subgroup. For example, if you're doing a t-test, I'm sure some people have heard of a t-test, you can fit a t-test in men and a t-test in women and compare the results. Or you can use an interaction term in a model. Paul just talked in depth about an interaction term. If you were looking at, say, treatment and gender, you would have a model with an interaction between treatment and gender. That's another way to do a subgroup analysis of gender. That's really all I'm going to talk about for the subgroup analyses. Before I do open it up to questions, I do want to say that in the slides that were provided to you before the workshop, there are all the references that we referred to, as well as additional ones that can be useful for you. So please take a look at those when you have a chance. And now, if we can please open it up for questions.
And if you do have a question, press star then 1 on your touch-tone phone.
I mean, I just want to, if you have questions about what we discussed, of course, we'll be happy and maybe we'll take those first. But we have the luxury of having plenty of time for Q&A, and you've got two biostatisticians here. So if you have questions on things we did not cover, we'll try to answer them. We can't guarantee that we'll know the answer, but we'll definitely try. So either relevant to what we discussed, or really besides other topics that we did not discuss. If you have questions on any of those, feel free.
Question from Therese Kylene. Please go ahead.
Hello?
I hear you now, Therese.
Well, I think I had pressed it before. Actually, I had pressed it before the last presentation. and it was just kind of a follow-up to what I had asked before. If you're in the grant, you write other psychosocial outcomes will be evaluated in subgroup, or I'm sorry, other psychosocial outcomes will be evaluated. If you write, does that cover specifically, or do you have to? Or would it be better to cite exactly what kind of biopsychosocial outcomes will be explored in a secondary analysis?
But do you know what they are?
Well, a lot of times it's ASI subscale scores, but it could be BDI, other substances of abuse. But you see, often in protocols, that's how it's worded.
But I think if you know what they are, I would list them. I would really list them. And again, if you list them and you say, I realize there are a lot of them, but they're all important and they're worth exploring. And yes, they're not going to be the definitive answer. So I think, how do you say it? Transparency and honesty goes a long way. And if you don't know them, if you don't have, you really mean other, like, I'll see what I can do, then, yeah, they really should be taken with a grain of salt. Does that answer, or Abby, anything you want to add? No?
Yeah, no, I had just seen, I saw that in some studies, so just thought I'd bring it up.
Yeah, well, thank you for your question.
Once again, if there are any further questions, press star then 1 on your touch-tone phone. We do have a question from Nahima Ahmed. Please go ahead.
Hi. So when you're talking about subgroup analysis, do you think it's better to do them as a separate analysis or to control for them in your model?
Oh, the different ways of doing it analytically? It's tricky. I don't know that there's a clear preference. I think, well, I know Paul has an opinion.
Yeah, I do have a preference. I think, to be honest with you, I can't tell you it has statistical, I think it does, but I'm not 100% sure. But yeah, I would use the interaction, because what the interaction is, it's basically saying, yes, the treatment effect is different in your subgroups. So when you do a gender by treatment, and a gender by treatment interaction, and you put a gender by treatment interaction in your model, it's a one test, that tests for the interaction. So it's a one test on the interaction, and the answer is yes or no. It's significant or it's not. And so this is a more direct way of testing whether the two groups are different. The other way, which is of course is valid and all that, but the other way says, I'm going to test for males and I'm going to test for females, and I'm going to get 2 p-values. Now look, you already have 2 tests now instead of just 1. And of course gender is easy because you only have 2 groups. What if you have 6 or 7? So now you're going to do 7 test and get 7 p-values, and some are going to be significant, some are not. Maybe all are, maybe all are not. And but if you have 6 or 7 categories and you put just the interaction term, the model is going to tell you, yes, there is a difference between your subgroup. That's all it answers. And then you dig down further to find out in what way they are different. So, I mean, again, I think it is statistically better, but I can't tell you 100% sure that it is. But that would be my preference. I don't know. Abby.
Yes, I completely understand what Paul is saying. However, one of the issues is power. And when we do relegate our subgroup analysis to looking at an interaction, the problem is that in a model, testing for the interaction term is going to be woefully underpowered. So the fact that you don't reject means less than if you didn't reject and it was some other term in the model. Because if you have, like, say, main effective treatment and main effective site and then the interaction, for power, that interaction term has less power than the other tests. So that's important to keep in mind when interpreting the interaction term for the subgroup analysis. So that's kind of why I was leaning towards, there's not one clear, real answer. But again, if you do the interaction term, yes, it does have this nice, simple test, and it leaves it much less open to interpretation. However, it does have the drawback that it can be underpowered.
Right. Absolutely. Very, very good point. And, while Abby was talking, I thought about 2 things. One, we're not talking about putting the interaction in the primary analysis. We're talking about, in the secondary analysis. So you don't want to fill your primary model with all kinds of interactions. So that's number 1. Number 2, absolutely, Abby's absolutely right. When you do the separate ones, you can see, you remember those confidence intervals that I show in the basic trial design? Well, now you can actually picture them, and let's say you put the male and the female on a graph, and you can see, how far apart, whether it's clinically meaningful, the difference between the two. So yes, when you do each separate, it does have advantages of actually seeing it has more clinical meaning than just the p-value of the interaction term. Yeah, I agree.
Yeah, so something that, I've explored is, yes, you do the model with the interaction and you test for it, but you also look at it graphically as much as possible because of this fact that you might have a power issue. And, it's not a black and white issue. We can't say, oh, there's definitely a treatment by gender interaction. If you reject the test, but you look on a graph and the 2 groups clearly have different treatment effects or even different directions, it's just one of those things that makes statistics very much an art and not as much a science.
And if no one else has another question, I want to ask about analysis plans, if that's okay.
Sure, go ahead.
So the other question that I have, I know that as much as possible you want to put in what tests you're going to run. But before you have collected data, should you assume that all the assumptions of your test will be run, or is it better to say that I'm going to use a non-parametric test because I don't know how the data will be distributed?
I mean, one thing I have done in protocols, not actually for NIDA, is that I've seen this in other projects as well, is you have a contingency plan in your analysis plan. So you say, yes, I'm going to, for example, do an ANOVA, which makes a certain assumption called normality. But if we have substantial evidence that assumption doesn't hold, we're going to do test B instead. And that is one possibility. You can specify these alternative options if certain assumptions do not hold. The only problem with doing that you run into is when you power your study, you have to pick one. And that's the really tricky part. Do you have anything?
No, I completely agree with you. I mean, that's how I would have answered. I mean, it's a very good question because we don't know. I mean, we can't, know exactly the distribution of the outcome, the distribution of this, the assumption of that. So, but Abby said it very well, which is, there's nothing wrong with saying, I'm going to test this first, and if it doesn't look like things are normal, well, I'm going to use non-parametric, and if it doesn't blah, 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 I'm going to do this third option. There's nothing wrong with that. But again, Abby's point is a very good one in terms of, for the sample size and all that, you've got to pick something.
Yeah, as opposed to immediately jumping into always using a non-parametric test, they are generally conservative. So it can't be your general policy that I'm always going to do a non-parametric test. I'm just going to hedge my bets. Because, for example, if you did have normality, then your test that assumed that would be more powerful than a non-parametric test. So that's why people don't just always use non-parametric methods.
Thank you.
We have no further audio questions at this time.
Okay, great. Well, at this time, the CCC would like to thank everyone for participating, and particularly to our presenters, Abigail Matthews and Paul Joaquin. This was a very engaging discussion, which will be available on the CTN Dissemination Library in the upcoming weeks. We'd also like to encourage others to please complete the post-webinar survey. That way we'll get your feedback. And like I said, and also if there are other questions, or if you were just too shy today to chime in, we really, really would like to hear from you, and Paul and Abby are available for additional questions, particularly if it's specific to your protocol. Please email us at ctnsupport@ms.com or ctntraining@ms.com. That's at emmes.com. We would be happy to pass that information along. Thanks, everyone, for participating. Our next webinar will be Wednesday, December 19th, on helping patients with substance use disorders and pain, which will be presented by Jennifer Sharp-Harder and Roger Weiss. Thanks, everyone, and have a great evening.
Thank you, ladies and gentlemen. This concludes today's call. Thank you for participating. You may all disconnect at this time.